Hello, my fellow colleagues. My name is Andrew Alexander. Hello, my fellow colleagues. My name is Andrew Alexander Martini, and today I will be presenting for this week's discussion topic. I will be talking about possible stem cell related therapies by taking a detailed look into this article I found on neural stem cell transplantation and its effects on decreasing neuroinflammation in a transgenic mouse. So before we get into the paper, I would like to talk about some general facts about Alzheimer's disease. A very basic definition is shown in the slide: an irreversible, progressive brain disorder that slowly destroys memory and thinking skill, and eventually the ability to carry out even the simplest tasks. Onset of this disease can occur as early as someone's 30s or as late as someone's mid-60s. Symptoms of early stages of the disease include forgetting important things like dates and newly acquired information, repeated questions over and over again, basically asking the same question over and over, losing track of time, misplacing things, increasingly poor judgment, as well as changes in mood and personality. As time goes by, these symptoms get a lot worse, and some of these later stage symptoms of the disease include deepening confusion about time, place, and life events, severe memory loss, and suspicion about friends and family, not being able to recognize your loved ones and who they are and what they mean. So I just wanted to give you guys some general information about what Alzheimer's disease is, because the paper does investigate this specific neurodegenerative disease, Alzheimer's. So to get things started, I would like to go over some background information concerning what the authors were trying to investigate and why. So it has been shown in a number of studies that the inflammatory process is important in the progression of neurodegenerative changes in Alzheimer's disease and is highly correlated with cognitive deficits in AD-like mice. Because of these findings, transplantation of neural stem cells has been considered as a potential new therapy for the treatment of AD because of its effects in improving cognitive ability, which is something that is lost as AD progresses. So neural stem cell transplantation has been considered as a potential therapy. However, the protective effect against inflammatory changes of NSCs has not been looked at and must be evaluated. So in this study, they are looking for proteins and specific markers to measure that relate to these inflammatory changes already described. These markers/proteins include glial fibrillary acidic protein, ionized calcium binding adapter molecule 1, and toll-like receptors 4. Also quantified were TLR4 pathway-related mediators, A-beta concentration, and pro-inflammatory mediators. So the quantification of these markers/proteins is going to help analyze cognitive function in the transgenic mice that the authors have selected. In this slide, I want to just cover a little bit more background before we get into the materials and methods, because I would really like to briefly talk about microglia and astrocytes, as they are important. Microglia and astrocytes are neuroglial cells which provide homeostatic support, protection, and defense to nervous tissue. Microglia are responsible for removing damaged neurons and infections, as they are a type of macrophage, while astrocytes are responsible for glutamate, ion, and water homeostasis, defense against oxidative stress, energy storage, mitochondrial biogenesis, scar formation, and tissue repair. It has been shown in a number of studies that microglia and astrocytes have an important role to play when it comes to AD progression. Reactive microglia and astrocytes have been seen around amyloid plaques, which are misfolded proteins that form in the spaces between nerve cells. These reactive neuroglial cells express toll-like receptors, like mentioned before, and have build-up responses to innate immune triggers by releasing pro-inflammatory molecules. Other observations of glial cells have come to suggest that glial cells and TLR4 signaling pathways are important susceptibility factors for Alzheimer's disease. Now let's put all this information together so we can get a good general look at what we are going to be investigating. Inflammatory processes have been highly correlated with cognitive deficits in AD. Neuroglial cells have been seen around amyloid plaques. These glial cells release pro-inflammatory molecules and mediators, and contribute to the development and progression of cognitive deficits in AD. One of these mediators is TLR4, which is closely associated with the inflammation that occurs in AD pathogenesis. So, the study is basically focusing on the clarification of these neuroinflammatory mechanisms involved in their NSC transplantation strategy. I also provided a general workflow and order in which these techniques take place during the experimentation process. Going from the top to the bottom, first is the generation of EGFP-labeled neural stem cells, followed by transplantation into recipient mice. Behavioral training via the Moore's water maze test was done next, followed by blood sample collection, identification of all three neural lineages, neurons, astrocytes, and oligodendrocytes, / immunohistochemistry, as well as RT-PCR, Western blot, and then ELISA. Here I have listed a number of techniques used for further analysis of cognitive deficits via measurement of markers / proteins that are important in AD pathogenesis. These include the types of animals used, EGFP-labeled NSC generation, transplantation, and identification in vivo, the Moore's water maze test, blood sample collection, immunohistochemistry, RT-PCR, Western blotting, ELISA, and statistical analysis. The animals used in this study were APP/PS1 double transgenic or TG mice and age-related wild-type WT littermate males. All mice were maintained under a 12-hour light/dark cycle with food and water. This slide shows the generation and transplantation of neural stem cells. The neural stem cells were harvested from day 14 embryonic non-TG B6C3 mice. These cells were cultured in T25 tissue culture flasks in Delbecko's modified Eagles medium, which consisted of neurobasal medium, 2% B27 supplement, human recombinant epidermal growth factor, fibroblast growth factor, and heparin. The cells were cultured at 5% CO2 at 37 degrees Celsius as a suspension culture. The NSCs were then transfected with the lentiviral enhanced green fluorescent protein at passage 2 and transplanted into recipient mice at passage 5 after being sorted by flow cytometry. Following the materials and methods list, this is what is known as the Moore's water maze test. Acquisition of spatial learning began 10 weeks post-NSC transplantation for 4 consecutive days. The task consists of a pool of water situated in a room with salient cues. There is a platform located 2 centimeters below the water surface, and mice are allowed 60 seconds to locate the platform. If the mouse was unable to locate the platform, they were manually guided to it and remained there for 10 seconds. The mean escape latency per day was recorded for each animal and used in statistical analysis. One day after the final acquisition training session, all mice performed a probe test with the escape platform removed. The percent time spent in the target quadrant where the platform was before was recorded and interpreted as a representation of spatial memory. The authors are basically trying to see if the mice can attain spatial memory based on remembering the location of the platform. After completion of the behavioral testing via the Moore's water maze test, all mice were deeply anesthetized and blood samples were collected. Blood samples were centrifuged at 1250 G for 10 minutes at 4 degrees Celsius. The resulting supernatants were stored at -80 degrees Celsius for further analysis. After blood sampling, the brains were removed from the skull and sectioned longitudinally for tissue processing. So below I've included a picture of just general immunohistochemistry protocol for staining tissue. The antibodies used were specific to MAP2, GAL-C, GFAP, IBA-1, A-beta, and TLR4. Brain sections were treated with 0.25% Trident X-100 for 10 minutes and then blocked with 10% donkey serum. Sections were then incubated overnight at 4 degrees Celsius. The next day, after washing, the slides were incubated at 37 degrees Celsius for 2 hours with a secondary antibody, followed by incubation in Hoist 33342 for 10 minutes. Images were taken with a laser scanning confocal microscope. The expression of GFAP, IBA1, and TLR4 mRNAs from NSC transplanted AD mice were examined using real-time PCR, or RT-PCR. The total RNA was isolated from frozen cortical samples with triazole, according to the manufacturer's instructions. RT-PCR of cDNA was performed using the forward and reverse primers listed in the slide. As you can see, there are forward and reverse primers for GFAP, IBA1, and TLR4. The GADPH was used for data analysis. Western blotting was also done to visualize bands of target proteins GFAP, IBA-1, and TLR4. SDS page was conducted on polyacrylamide gels, and proteins were separated and then transferred to a nitrocellulose membrane. 5% bovine serum albumin was used as blocking solution for 1 hour at 25 degrees C and immersed overnight at 4 degrees C in the following antibodies: anti-GFAP, anti-IBA-1, anti-P-P38 MAPK, or anti-P-nuclear factor kappa, P65, or in anti-TLR4, anti-myeloid differentiation primary response gene MYD88, or anti-TIR domain containing adapter inducing interferon-beta. Horseradish peroxidase labeled anti-rabbit or anti-mouse secondary antibodies. Afterwards, the membranes were washed and enhanced chemiluminescence was used to visualize the bands. Finally, the levels of inflammatory mediators and soluble and insoluble A-beta in the brain of AD mice were quantified. Frozen brains were weighed and homogenized in tris-buffered saline containing a cocktail of protease inhibitors. Then the homogenates were suspended in 2% SDS and centrifuged at 100,000 G for 1 hour at 4 degrees C. Supernatant was collected for the soluble A-beta ELISA, and the remaining pellet was dissolved in 70% formic acid and centrifuged again. The supernatant was collected for the insoluble A-beta ELISA. Commercially available sandwich ELISAs were used to quantify protein levels of A-beta as well as inflammatory mediators. Absorbance at 450 nm was measured on an ELISA reader. So the results of the experiment are as follows. We have 3 graphs all showing how transplantation of the NSCs rescued cognitive deficits in the AD transgenic mice. To the left in graph A is the evaluation of spatial learning using the Morse water maze latency test at 10 weeks after transplantation for 4 consecutive days. The solid gray line represents the wild-type mice who do not have AD. The dotted line represents TG vehicle mice, meaning they have AD but do not contain transplanted NSCs, and the semi-dotted line are the transgenic mice that have AD but have been treated with transplanted NSCs. In the graph, there's a significant decrease in latency during which the mice must locate the platform in the TG NSC mice compared to TG vehicle mice on days 3 and 4. This could indicate an improvement of impaired spatial acquisition. Looking at graph B and C, during the spatial probe test, TG NSC mice spent more time in the quadrant and crossing the quadrant where the platform was located than TG vehicle mice, also showing effective attenuation of spatial memory deficits in AD mice. Here is a graph that shows engrafted neural stem cells that have migrated and differentiated into three neural lineages. Photos A through D show EGFP positive NSCs surrounding the hippocampal injection site. Photo B are engrafted NSCs found within the CA1 region of the hippocampus. Photo C shows the NSCs in a subfield of the hippocampus and also within the overlying corpus callosum. And finally, photo D shows the NSCs migrating into the cerebral cortex. Photos E through G show NSCs that have differentiated into neurons, and the way you can tell is by the co-expression of MAP2, which is labeled in red. Photos H through J show NSCs that have differentiated into astrocytes, shown in red, co-expressing GFAP. And photos K through M show NSCs that have differentiated into oligodendroglia, co-expressing GALC, also shown in red. So basically, the transplantation and differentiation of these neural stem cells into the hippocampus and migrating up toward the cerebral cortex was a success based on the confocal microscope images presented here. Accumulation of A-beta deposits is a characteristic feature of AD neuropathogenesis. Since the initial description of such amyloid plaques, reactive microglia and astrocytes have been observed around these plaques, as indicated by IBA-1 and GFAP expression in the AD brains. Hence, the immunofluorescence experiments were designed to assess whether engrafted NSCs affected glial expression around amyloid plaques. Images A through C show the number of microglia and astrocytes surrounding A-beta plaques in the AD brain at 10 weeks post-transplantation. To break it down further, photo A shows images of A-beta staining, shown in red, and activated astrocytes, shown in green, in wild-type mice. Photo B shows the same representative staining, but in TG vehicle mice, and photo C shows the same staining for TG NSC mice. From left to right, photos E through G shows representative images of A-beta staining in red and activated microglia in green in wild-type, TG vehicle, and TG NSC mice. In the TG NSC and TG vehicle groups, microglia and astrocyte proliferation typically appeared to surround the plaques. For the wild-type group, there were no A-beta plaques observed. Finally, photos D and H are quantitative analysis showing significant group differences between the TG NSC and TG vehicle mice groups. As you can see, the area occupied by ABA-1 and GFAP are significantly lower in the TG NSC than they are for the TG vehicle group. There are less activated microglia and astrocytes in the TG NSC compared to the TG vehicle mice group. These graphs show the quantification of A-beta peptide levels in the brain and plasma of AD mice. In many AD transgenic models, A-beta peptide plays a key pathogenic role in AD. Thus, in order to understand the effect of engrafted NSCs on beta amyloidosis, A-beta 40 and A-beta 42 were measured to analyze and determine A-beta levels. Graph A shows the soluble A-beta 40 and A-beta 42 levels in TG vehicle versus TG NSC mice groups, whereas graph B shows the insoluble A-beta 40 and A-beta 42 levels in TG vehicle versus TG NSC mice. Analysis of both soluble and insoluble levels of A-beta 40 and A-beta 42 via sandwich ELISA showed no differences after transplantation. Finally, graph C shows the analysis regarding level of A-beta 40 and A-beta 42 in plasma, which also show no difference after transplantation. Thus, NSC transplantation rescues cognitive performance in AD mice without altering A-beta levels. Here are a couple more images and graphs relating to activation of glia and TLR4 in AD mice. In images A through F, double immunofluorescence analysis was done to show the activation and localization of GFAP IBA1, and TLR4. Photos A and D are for wild-type mice, B and E is for TG vehicle mice, and C and F is for TG NSC mice. Looking at image C, we can see that the levels of GFAP shown in green and TLR4 shown in red are decreased in the TG NSC mice compared with the TG vehicle mice located on image B. Looking at images D through F, we can see a decrease in expression of IBA1 shown in green and TLR4 shown in red in the TG NSC group shown in image F compared to the TG vehicle group shown in image E. Graphs H through J below show relative expression levels of all three proteins via RT-PCR between all three mouse groups. Looking at the TG-NSC-AD mice, we can see a significant drop in GFAP, IBA1, and TLR4, showing decreased glial activation. Western blot analysis can be seen on gels G and K as the levels of GFAP, TLR4, and IBA1 were lower in TG-NSC mice compared to TG vehicle mice. Graphs L through N also show the same result, decreased levels of GFAP, IBA1, and TLR4 in the TG-NSC groups as compared to the TG vehicle groups. Overall, decreased expression levels of these three proteins means decreased glial activation, which means decreased mediation of pro-inflammatory pathway molecules. Here is an analysis of TLR4 pathway-related agents MyD88 and TRIF. P38 MAPK and NFKB P65 are also important as they are involved in MyD88 dependent or TRIF dependent TLR4 events. Looking at the Western blot analysis on the top left, we can see that expression of MyD88, TRIF, P38 MAPK, and P-NF-KB P65 were lower in the TG-NSC compared to the TG vehicle mice groups. Looking at the graphs showing relative expression of all four TLR4 pathway-related agents, the TG-NSC mice show lower expression of all four pathway-related agents compared to TG vehicle mice. And so, once again, we can see a pattern here. Decreased pathway-related agents means decreased TLR4 activation, means decreased glial activation, and decreased pro-inflammatory processes, and increased cognitive improvement. As I mentioned in the previous slide about decreased pro-inflammatory processes, here we examine expression levels of pro-inflammatory mediators in AD mice. Levels of pro-inflammatory cytokines in both brain, shown in graphs A through D, and plasma, shown in graphs E through H, are decreased for the TG-NSC mouse group compared to the TG vehicle group. In wild type, the expression of these cytokines is even lower. The pro-inflammatory factors investigated in brain and plasma include interleukin-1 beta, also known as IL-1 beta, interleukin-6, tumor necrosis factor alpha, and prostaglandin E2. And once again, looking at the graphs, the same pattern emerges. Decreased pro-inflammatory mediators means decreased TLR4 pathway activation, means decreased glial activation, which leads to decreased neuroinflammation. So what does this all mean? As has been stated, inflammation plays a critical role in AD pathogenesis, and recent studies have indicated that NSC transplantation can serve as a potential treatment for AD. However, the mechanism by which neuroinflammation is triggered in the NSC-treated AD brain is poorly understood. Thus, in this study, the mechanism of neuroinflammation was explored, since in AD, the prefrontal cortex of the brain is the region that loses the greatest number of neurons. Exploration of neuroinflammation was done by measuring the expression of factors related to glial activation, such as GFAP, IBA1, and TLR4. This phenomenon was studied further in depth by looking at expression of factors that contribute to TLR4 activation and pro-inflammatory cytokine expression, which are pro-inflammatory mediators present during TLR4 pathway activation. So here is a schematic diagram depicting the possible mechanism for neural stem cell transplantation decreasing neuroinflammation in the AD mice. Transplantation of NSCs is done through the hippocampal area of the brain in the AD model transgenic mice. Thus, microglial and astrocyte activation is decreased, which leads to a decrease in TLR4 signaling pathway. This leads to a decrease in inflammatory mediators, which then leads to a decrease in neuroinflammation of the affected region, which in turn helps restore some of the cognitive deficits that is seen in Alzheimer's disease. I would like to take this time now to critically evaluate the study in question. I feel as though the methodology and steps taken to show cognitive improvement in the AD mouse brain was well designed and thought out. Exploring the many factors that contribute to neuroinflammation was a good strategy as it provides quantitative data of proteins and molecules that are related to neuroinflammation and contribute to AD pathogenesis. One of the concerns that I have regarding this study is the fact that mouse neural stem cells were used instead of human neural stem cells. Could there possibly be a difference in the neuroinflammation mechanism if NSCs from human are used instead of mouse? Another concern I have is the amount of behavioral training done on the transplanted AD mice. Is 10 days enough time to really evaluate cognitive ability? If the mice were trained for 20 days instead of 10, would the results be similar or different? Since activation of microglia and astrocytes leads to increased levels of GFAP, IBA1, and TLR4, would we see increased expression of these factors in neurons surrounded by the microglia and astrocytes? Is depolarization and neuronal firing affected by increased expression levels of these factors? And would this cause the breakdown of the neuronal cytoskeleton? All these questions and factors that I felt would be important to address and perhaps study in the future. Thank you very much for taking the time to listen to my presentation. I hope you all enjoyed the topic in question, and if you have any further questions, please post in the discussion forum. Thank you.